lots of chatter over there. Thank you everyone for engaging and letting us know who you are and where you're at. I am Sarah Fitzsimmons. I work for the American Chestnut Foundation. I'm the Chief Conservation Officer. I am in my lab here at Penn State University in State College, Pennsylvania. And I'm joined today by Jonathan and Camila from the University of North Carolina in Asheville. And so before we turn it over, I want to thank them again for coming, and for Vasily Lakoba, who is not here. Vasily is actually driving on his way from Meadowview, Virginia, up to Central Pennsylvania. He is going to be giving a talk at the Pennsylvania, New Jersey chapter meeting tomorrow at the historic Annville Theater in Annville, Pennsylvania, just east of Hershey, PA. So if you've ever wanted to visit Annville, tomorrow would be a great time to do that. Join us for the Pennsylvania, New Jersey chapter there. I'll be there, like I said, as will Vasily. But Vasily made this happen with Jonathan and Camila. Thank you again for joining us. Before we kick all that off, a little bit of housekeeping. Number one, if you are not me, Camila, or Jonathan, we can't see you, we can't hear you. So if the dog barks, if you want to have lunch, if you want to watch Price is Right at the same time, go ahead and do that. It's not going to bother us. You go about your own day, and we'll just keep on talking over here. If you have a question, there is a Q&A module. So if you scroll down to the bottom of your Zoom screen, you'll see a little box that has a question mark on it, and it says Q&A. Throw your questions in there, and we will keep track of that as time goes on. I also have an option where you can upvote questions. So if you see a question that you really like, give that the thumbs up, or also you're welcome to comment on questions and answer them yourself. Feel free to engage as you wish. If you put a question in the chat, I may lose it because I'm basically solo this time. Usually I have someone who's monitoring the chat and the Q&A. I don't have that today. So if you put a question in the chat, I may lose it. Put your questions in the Q&A, put your witty banter and weather reports in the chat. Next month, April 18th, we will return to our third Friday schedule. I'm going to do a town hall, an open town hall for chestnut growing. So bring all of your questions about how to grow American chestnuts. If you want to share a quote unquote hot tip on how you grow chestnuts, or protect them from critters, or make them grow faster, or whatever, get in touch with me. I am offering up an opportunity for you to be a panelist, one of the esteemed guests on Chestnut Chat. If you have some cool pictures and a cool story you want to tell about what you do to make your chestnuts grow amazing and gorgeous, let me know, and I'll include you in the panelists. Otherwise, it'll be open question, and we're just going to have staff and longtime growers talking about how to grow chestnuts and answering any of your questions that you may have. I think that's it for all the housekeeping. If you need anything, like I said, statements of fact or other witty banter in the chat, specific question for Jonathan or Camila, throw that in the Q&A. Jonathan's going to go first. He'll give his talk. If you have a really big question, if you don't understand something, I think, Jonathan, you've said that you are willing to be interrupted. That's totally cool. So throw that in the Q&A. I'm going to keep watching that. I will interrupt Jonathan as he goes through if you have a question to try and get him to clarify. Otherwise, we'll leave all the other questions to the end. Have a little bit of a mini Q&A between Jonathan and Camila. Then we'll switch over and then have a longer Q&A at the end. Sound good? All right, fantastic. Well, without further ado, Jonathan, I'm going to turn it over to you again. Thanks for joining us, and take it away.
Yeah, thanks for the invitation, Sarah. And thanks for facilitating this. So I'm Jonathan Horton. I'm a professor and chair of the biology department at University of North Carolina, Asheville. And Camila and I received a small grant from the American Chestnut Foundation to do the work that we're going to present today. And so all of this work that we're going to talk about was done by our undergraduate students here at UNC Asheville. And I'm trying to resize all of these little boxes from Zoom so that they're not blocking my view. So pardon me while I do a little housekeeping. Okay, I guess that's as good as I can get it. So anyway, what I'm going to talk about today are the physiological characteristics and a comparison of pure American chestnuts with some chestnut hybrids. And so some of this is basically a talk that my student, Sam Cleary, put together as part of his undergraduate research, and he graduated last spring. So some of this will be very basic review, right? You all know the American chestnut was a dominant keystone species. It was up to a quarter of the canopy coverage in the southern Appalachians before its demise. It was very ecologically and economically important. And then the introduction of the chestnut blight basically made this species functionally extinct on the landscape. And so this fungal pathogen spreads pretty quickly through spores, and it creates these cankers that you can see in this image on the right that basically girdle and kill the tree. And so, as you know, since you're all members of the American Chestnut Foundation, there's been an effort by the foundation to try to develop blight-resistant American chestnuts to restore this foundation species back to the landscape. And so this is just an overview of the breeding program that you all know. And so what we're going to be presenting today is some physiological data from pure American chestnuts, and then the B3F3, so the 15/16 chestnut, and then the first back cross, so 3/4 American chestnuts. So these were all planted at DuPont State Recreational Forest, which is about an hour south of Asheville, kind of near Brevard. It's actually where there's some fires happening right now. And so these were planted from seed collected along the Blue Ridge Parkway for the 100% American chestnuts, and then seedlings that were grown by the Chestnut Foundation that were, I guess, extra after some of their uses. And so these were planted in different stands very close to each other, within a kilometer of each other, in sort of the eastern part of DuPont State Forest. And so we were really interested in looking at the performance of these trees in sort of a natural forest environment. And so we're hoping that this information will help us inform future restoration efforts. So the first thing we did is we used this fancy piece of equipment here. This is a portable photosynthesis system made by Lycor Lincoln Biosciences in Lincoln, Nebraska. And so you can see this little cuvette up here. We clamp this on a leaf. And within that cuvette, we can control the environmental conditions. We can control the light. We can control the CO2 levels, the temperature to some degree, the humidity to some degree. And so this measures plant photosynthesis, so the amount of carbon that the leaf is taking up, but also transpiration, the amount of water vapor that's given off by the plants. And so the first thing we did was to use this instrument to construct what we call steady state light response curves. And I'll explain that in a minute. But this is a diagnostic tool to assess a variety of photosynthetic characteristics and to compare them among these three different types of chestnuts. And so in late July of 2023, my students and I were down and we chose 6 trees per site that were all under consistent light environments. And then we used this instrument to measure a steady state light response curve. So what you do is you set the light in the chamber to your highest point, clamp it on the leaf, let the leaf equilibrate to that, and then you measure photosynthesis, and then you step it down through 10 or 15 different light levels all the way to darkness. And we did all these measurements in the morning or before 2 p.m. to avoid late afternoon heat stress, which can cause the plants to kind of shut down. And so this is what a light response curve looks like. So you can see at our highest light level, which is about 60% of full sunlight. And we have the American, the 100% American, the 94%, and then the 75%. So this 75% American has 25% of the Chinese chestnut genes. And one thing that just pops out at you looking at this is that there's a pretty good similarity between the 100% American and the 94% or 15/16 American, which is sort of the target for reintroduction. And so that's really good that it's behaving similarly physiologically to the 100% American chestnuts. And then you can see the trees that have the higher component of the Chinese chestnut have different photosynthetic characteristics. And so the parameters that we can get by fitting a model to these light response curves, one of which is the maximum photosynthetic rate. So this is the highest photosynthetic rate that the trees can get. So you can see that basically the 94 and the 100% have almost twice the photosynthetic rate of the 75% American. Another thing that we can get from this is a measure of the dark respiration. So all living things respire. That's how we get our metabolic energy. A lot of people think trees photosynthesize, animals respire, but plants respire also. And so the reason that we measure this photosynthetic rate in the darkness is to get a measure of that dark respiration, that maintenance respiration rate. And you can see there's some differences among those also. Quantum yield, this is a little bit technical, but this is as light increases in this initial part of the curve, photosynthesis will increase. And so it's a measure of how quickly photosynthesis increases with increasing light. And then lastly, the light compensation point is where this net photosynthesis is 0. And so that means that the plants are gaining enough carbon through photosynthesis to counteract the carbon that they're losing through respiration. So this light compensation point is sort of a measure of shade tolerance. If they have a lower light compensation point, they can persist longer in shade. And so these are all really important parameters to kind of understand how plants photosynthesize and how they can react to different environments. And so one thing that you can see, like I said, is that the 94% and the 100% were very similar, and they have higher photosynthetic rates at these higher light levels than the 75%. And if we look at these parameters, the mean parameters among the groups, you can see here that the maximum photosynthetic rate was very similar. It was not statistically significantly different between the 100% and the 94%, but was significantly lower in that 75%. And then looking at the dark respiration rate, this is kind of interesting because the 94% had a much higher dark respiration rate, which is something that we weren't expecting. We were expecting it to be more similar to this. The quantum yield was also higher in the 94%. So generally, plants that are more shade tolerant tend to have higher quantum yield. And so this is kind of an interesting finding that it's kind of the highest quantum yield was in the 94%, lowest in the 100% pure American, and then intermediate in the 75%. So I'm still not sure, I don't have a good explanation for why that is, but this suggests that these 94% may still have some of the more shade tolerant characteristics of those Chinese chestnut genes. And then this light compensation point kind of ties together the respiratory cost and the quantum yield. And so even though the 94% had a much higher maintenance respiration because it's more efficient at using light, it had a non-significantly different light compensation point. So the light compensation points were all very similar. So those were the steady state measurements just to try to assess what the overall photosynthetic characteristics are. And this, interestingly, there was a paper done by some people in 2014 doing similar things with a broad range of pure American, pure Chinese, and then different backcross hybrids. And our ranking of the photosynthetic characteristics were similar, right? With the American being having the highest rates of photosynthesis and the Chinese having the lowest, and then the hybrids in the middle. Although the rates of photosynthesis in their study were a lot higher than the ones in ours, and that could be due to the ambient light environment or some other environmental factors that differ between the study sites, between our study site and theirs. But the ranking was very similar. So the responses of the trees were very similar. So the next thing we did is we wanted to look at how the plants photosynthesize during the day, but also respond to water stress during the day. So typically, in the afternoon, it tends to be hotter and drier, and so plants can experience water stress, and sometimes that can negatively affect photosynthesis. So plants have stomata on their leaves, or the pores through which they gain carbon and lose water. And if they start to experience water stress, they'll close those to conserve water, and that will decrease their carbon gain. So we're really interested in looking at these diurnal patterns throughout the day. And so we did this with, we did not include the 100% American in this study because, as you likely know, the 100% American chestnuts are very susceptible to blight. And so those trees had all been top killed and had resprouted and were very like seedling size or shrub size, whereas the other trees were a lot larger. And so we thought that comparing all three of those would not be valid because of the different stature of the three types. So we only looked at the 94% and the 75% for the diurnal patterns. And so we used this instrument on the right. This is what we call affectionately the pressure bomb, but it's a pressure chamber. And this is used to measure plant water potential, which is a measure of the plant's water status, how healthy the plant is. So we did that in the morning before dawn, which is when this picture was taken, because the best time to measure a plant's water status when it's at its peak is in the morning right before the sun comes up, because they get to equilibrate. They're absorbing water all night, so they're kind of at their healthiest right in the morning. So we do the pre-dawn, and then we did a mid-morning measurement, and then we did an afternoon measurement. And the mid-morning and afternoon measurements, we also did photosynthetic measurements. And we did this in July and August. And so with that light-core chamber, what we would do is we would measure what the ambient conditions are that the leaf was experiencing and then set the chamber for those conditions. So when we were measuring leaf photosynthesis and transpiration, it was under the same conditions the leaf was on before we put the leaf in that chamber. So we're trying to measure like what the plant was doing at that time. And so some key findings from this, we also measured soil moisture. And so interestingly, even though these sites are less than 1 kilometer apart, there was a higher soil moisture in the 75% stand than in the 94% stand. And that allowed those trees to have a higher pre-dawn water potential. So water potential is kind of a weird thing to get your head around. It's a negative number, and the more negative means more stress. So this minus 0.55 means these trees had a lower water status than these trees in the morning. And then in the mid-morning gas exchange measurements, there were really no differences to see. And that's because morning is the prime time. They're all, very, very happy. But in the afternoon, when they're starting to experience that, late afternoon heat and greater evaporative water demand, you can see that the water potential declined in both of these to a similar state. So both were experiencing afternoon water stress. Even though they had very similar water potential, the photosynthetic rate of the 94%, so the trees that had the more American chestnut genes, had a much higher photosynthetic rate, even under the same level of water stress. And they had a slightly higher stomatal conductance, so that's a measure of how open or closed their stomata are. And they were also transpiring a lot more water. So even though they're experiencing the same level of stress, these trees that were more American seem to be performing better. And then these graphs to the right, this is where it gets kind of technical. And so if you have questions about this, let me know. So this is a measure of stomatal conductivity, or how open the stomata are. And you can see that as the stomata open more, both photosynthesis and transpiration increase, and similar increases for both species, at least in July. Oh, I forgot I had these animated arrows. So in August, you can see that the pre-dawn water potential measurements were very similar between the two, but the mid-morning water potential measurements, the 75% trees, the ones with the more Chinese genes, actually were experiencing greater water stress in the morning. And in the afternoon, that switched. So these trees basically kind of maintained their water potential similarly to what it was in the morning, whereas the more American, the 94% trees, had a much lower water potential. So they were experiencing more water stress. And this is showing the change in the water potential from pre-dawn to afternoon. So a much more dramatic change. So experiencing more water stress. But despite that, they still had higher rates of leaf gas exchange. So they had higher stomatal conductivity. They had higher rates of photosynthesis. They had higher rates of transpiration. So despite the fact that they were experiencing almost twice as much water stress, they were able to still perform and maintain photosynthetic rates at a much higher rate than those that had the more Chinese genes, which is pretty fascinating. And so again, these are the kind of the technical graphs. This is showing that stomatal conductivity again, so how open the stomata are. And you can see that if we think less about how open the stomata are and more about how closed they are. So in the afternoon, when plants are experiencing water stress, they tend to close their stomata. And you can see that for the 75% or those trees that had more Chinese genes, as they closed their stomata, it reduced both photosynthesis and transpiration. Whereas with the more American, the 15/16ths, as they closed their stomata, it did reduce transpiration, but it didn't affect photosynthesis. So they were able to maintain higher rates of photosynthesis and gain more carbon while experiencing greater water stress because it didn't have as much of an effect on their stomata. And this is similarly showing a similar pattern. So this vapor pressure deficit, this is a measure, it's kind of a measure of how dry the air is. So the evaporative demand, which is what's affecting stomatal conductivity. And what's really interesting here is that as the afternoon evaporative demand water stress increases, it causes declines in transpiration and it causes the stomata to close in those trees that have the higher proportion of Chinese genomes than in the trees that have the higher proportion of American genomes. So it seems like the 94% American trees were able to tolerate that afternoon atmospheric stress and maintain much higher rates of photosynthesis despite that extra stress. And so there's a word that kind of describes that pattern of behavior. It's called anisohydric. And so with that, trees that are more anisohydric will tend to have a riskier strategy. So they will keep their stomata open more in the afternoon to maximize that carbon gain, even though it may cause greater water stress. It's kind of a risky strategy because eventually, if the water stress gets so low, it'll cause cavitation and embolism in their xylem, the water transport tissue, and it'll make it non-functional. And that's generally bad. And so these more American trees have that more risky strategy. They're more gamblers, and so they're opening their stomata more, even under higher water stress, to maximize carbon gain. And so that could be really good because that can help them grow more. Whereas the trees that had the more of the Chinese genes had a more conservative strategy. So isohydric means they try to maintain their water potential in their tissues at the same level throughout the day. And I'm going to back up again. So if you see in the morning, the water potential of the 75% was minus 1.2, and in the afternoon it was minus 1. So pretty close. So they're maintaining it relatively the same. So isohydric, whereas the more American had a much more dramatic decrease in that water potential because they had their stomata open more, allowing more water loss, but also allowing much greater rates of carbon gain. And so basically they have these differing stomatal behaviors, these differing strategies for managing carbon gain and water stress. And so the 94%, the mostly American trees behave much similarly to what people have observed in 100% American trees in other studies. And so that's really good because we want to reintroduce this tree. We want it to be morphologically as similar to the original American chestnut, but we also want it to be physiologically very similar so that it'll hopefully occupy the same role that those trees had before they were extirpated by the blight. So that was really good that we found that they had very similar behavior and similar to what other researchers had found. And so this is important because we can utilize these to help assist with breeding programs for those blight-resistant American chestnuts. And then we can also provide recommendations for planting conditions, so determining the best places where you could plant these chestnut hybrids. And then integrate this with ongoing research to help inform the American chestnut restoration efforts. And so there's been very little physiological work done on these different types of hybrids of chestnuts. So I think any contribution that we can give to the broader body of knowledge on how these different types of chestnuts behave physiologically under a range of conditions and under a range of geographic locations, I think will help contribute to the overall restoration effort. And so with that, I just want to acknowledge the funding from the American Chestnut Foundation, our collaborators and colleagues at DuPont State Recreational Forest, particularly Jordan Bluff, who was the former managing forester there, and then the other students, so Kat Brown and Evan Hausler, who are pictured here, and then my student Sam, and this is them eating breakfast, because when you're doing these diurnal measurements and you get out there at like 4 in the morning to measure pre-dawn and you stay until 4 in the afternoon, you have to cook food over camp stoves. And so before I stop, I do have another brief thing I want to talk about. I was fortunate enough to receive additional funding from the American Chestnut Foundation for a project that I'm going to be doing this summer with more students at the Meadowview Research Farm in Virginia. And so this project is assessing the mycorrhizal communities among these different types of chestnuts. And so we're doing this reciprocal transplant, and I'll explain that in a minute, to see if these different chestnuts support different mycorrhizal communities. So if you're not familiar with mycorrhizae, these are fungi, symbiotic fungi that grow on the roots of plants, and they help the plants get water and nutrients, and then they get carbon from the plants for food. And they're very, very beneficial, very important, particularly during seedling establishment. So when seeds germinate, those new seedlings often tap into these mycorrhizal fungi, and often those fungi are connected through these networks to the roots of mature trees. And it's been shown that those fungal networks can transport water and nutrients between mature plants and their seedlings, and that helps give those seedlings a benefit and helps them survive and grow better during that most difficult stage of the tree's life cycle, which is seedling establishment. And so we're really interested in seeing whether the genetic makeup of those different types of chestnuts influence the community of mycorrhizal fungi in the soil. So, like I said, it's very, very important for those seedlings to tap into these common mycorrhizal networks when they're young. It helps with their survival and with their growth. And so what I mean by a reciprocal transplant is at the Meadowview farm, they have a stand of 100% American trees. They have a stand of 100% Chinese trees. And then we're actually going to use a 50-50 hybrid stand. And we have seeds of 100% American, 50-50 hybrids, and 100% Chinese that we're going to plant in each of these stands. So reciprocal transplant means each type of tree planted in each stand. And we're going to plant these out and use them as bait. We're going to plant them out, let the fungi colonize them, and then pull them up and look at what's colonized. So it'll allow us to see what the communities are within these 3 different stands, but also whether there are different networks forming between the American and these 3 different types. So our hypothesis is that when you plant these 3 types of chestnuts in an American stand, those seedlings that are most genetically similar to the parent trees will have better networks, and they will perform better because the symbiotic fungi are more similar between those 2. And likewise, we would expect the hybrid to do better here, and then the Chinese to do better here. And we are also planting those same seeds in a sugar maple stand, because sugar maples are non-ectomycorrhizal. So we can monitor these to see if they grow less well because they're not tapped into those mycorrhizal networks. And so we're going to plant the seeds next month. And like I said, we're going to act as bait. We're going to let the fungi colonize them. And then we're going to harvest them in August to look at colonization rates. And then we're going to use molecular techniques to sequence the DNA from those mycorrhizal root tips so that we can identify the fungal taxa, so that we can assess what the community composition in each of those stands is. And we're going to compare those communities among the stand types and the seedling types within those stands And we're also going to measure survival, growth, and physiology to see if those mycorrhizal connections or lack thereof in the case of that control plot affect the seedling performance. And so I'm really excited about this. We're going to be using some new technology that we have at UNCA to do the DNA sequencing. And so I'm really excited about this project. I was just up at Meadowview about a week and a half ago, meeting with the facility and others to kind of plan this out. And so with that, I think I will stop and answer any questions that you might have.
Yeah, we've got some great questions in here. So I want to thank everybody for great talk. Jonathan, thanks so much for putting that together. And like I said, we've got some good questions in here. We'll take a few minutes to take these. And so everyone, if you do have a question, please put that in the Q&A module. I saw someone, the pancakes, the camp stove pancakes are getting lots of love, as they should. So I'm glad you included that. Matthew Huang asks, is there data on hybrid complexes like Japanese chestnut times American chestnut, or he didn't ask this, but I'm curious if you have any plans to look at other species.
Currently, no. And I have not seen a lot of work on those. I know that in Meadowview, they have, I forget what they call it, but they've planted a bunch of different chestnuts that are not just Chinese and American in a demonstration area there. But I'm more interested in the Chinese American hybrids because that's kind of the main focus for the reintroduction. And so I'm a forest ecologist. I think it'd be great to get chestnuts back in the forest. And so I've not done any work with any of the other chestnuts. The only other Castanea that I have any other experiences is there was a lot of Castanea pumila, the chinkapin at one of our sites, and those things are tasty to eat in the fall. And they're not affected by the blight. So,
well, I wonder if because they are a shrubby species, I wonder if they would be significantly more shade tolerant because that tends to be their niche. That might be interesting to.
That's a good question. Yeah, these were actually growing in fairly open areas.
Okay.
And I've seen them where I've seen them in the Southern Appalachians are usually kind of in rocky areas like around Linville Gorge and stuff. So I don't know. I don't know how shade tolerant they are. If it's more shrubby because of the habitat that they're in.
Yeah. Let's see. Clark asks, do the higher levels of photosynthesis equate to faster growth for the more American trees?
Yes, I believe so. And so one thing that has come about from the literature that American chestnut and the most American chestnut hybrids, those 15/16 hybrids, they're actually really interesting because they have shade tolerant characteristics in the seedling layer so they can germinate and grow in the forest understory and then persist there for quite a while. But when the canopy opens, like say a tree dies or you cut the canopy, because they have that capacity to utilize high light, they can grow really fast. And so there are a lot of trees like that. And the oaks that are related are very similar. They have what we call a sit and wait strategy. So they can persist in the understory. And most of the growth they're doing in the understory, they're growing in the roots and storing carbohydrates below ground. And then when the light opens, they grow up really quickly. And so it's really cool to see that chestnut, the American chestnut, has that characteristics. The Chinese chestnut, because it's a shorter tree and more branching, tends to have more just shade tolerant characteristics and maybe not as much of that ability to grow quickly with higher light.
That's really interesting. Yeah, I've seen a lot of. I have some of my favorite tree cookies, not the kind you eat, but when you take a slice across section of a chestnut, you can see that release event very dramatically. Where you release the tree, you cut down the trees around it, open it up to the sun, and we've seen an inch plus in diameter growth in a season.
Wow.
Once you release that, so yeah, that sit and wait is absolutely appropriate for the species. Let's take a couple more. Ann Best asks, are the chestnuts from Chinese hybrids toxic? No. Chestnuts, all members of Castanea, are edible. You may be thinking of horse chestnuts.
Horse chestnuts, yeah.
You don't want to eat those. Those are a member of the Buckeyes, which are now in the Sapendaceae and somehow maples. I don't know.
Oh.
So Dana Nelson asks, plots of regeneration curves with only one curve indicates no difference between 3/4 and 15/16. Is that correct?
Yes. So photosynthetically, those curves were, I mean, there's a little bit of difference in the way the curve shaped kind of in the asymptotic region, but the rates were not significantly different. And they both had the similar maximum photosynthetic rates. So, which was encouraging that the 15/16 hybrids react physiologically similar to the 100% that we're hoping to replace.
I'll take a couple mycorrhizae and then we'll switch over
Yeah.
to Camilla. Because
My
I'm
favorite
glad
topics.
I'm glad you threw that in there. So Brooke asks, regarding the mycorrhizal study at MetaView, how can using the 50/50 hybrids show which fungal response is more similar?
So we had originally planned on using the more like hybrids closer to the 15/16, but part of it was seed availability. And actually, a lot of the original breeding program to see if there are differences between the American and Chinese is to take American Chinese and then that first hybrid to see what happens. Like, do those hybrids react more similarly to the American parent or to the Chinese parent? And so part of it is constrained because of seed availability. And then part of it is kind of going back to the original sort of research plan before they started doing the back cross hybrids. And so, and this is just a pilot study. And so I'm hoping we'll find really good information that we can kind of build on for the future.
Yeah. Well, let's take one more from John Hempel. Hey, John, is there any evidence suggesting that trees of different species growing close together harbor different mycorrhizal species?
Yes. Short answer.
Very
And also
simple.
what's really interesting is there's a lot of evidence to suggest that the fungi are not necessarily discriminate. And so that those fungal networks, there's been a lot of work done by Suzanne Simard in the Pacific Northwest showing that these common mycorrhizal network can form between trees of different species. And so yes, and in some of my mycorrhizal work, we found that there's even, there's a seasonal dynamic. So the mycorrhizae that are on tree roots early in the season is different than what's late in the season. And also a successional stage as trees get older, they support different mycorrhizae. So there are some that are better at helping seedlings and some that are better at, tapping onto older trees. But then there are also, it's kind of like that whole symbiosis of mycorrhizal is a continuum. And so there are some that are more beneficial and there are some that are less beneficial. So it kind of depends on a lot on the environment and what the community is. And that's when before chestnut became functionally extinct, there wasn't a lot known about mycorrhizal fungi. And so this is kind of really exciting to kind of open that box of what's going on in the soil with the fungi to see if the different genetic makeup of chestnuts influence what is supported in the soil.
Well, Jonathan, if you were trying to get back onto Chestnut Chat and to do a talk about mycorrhizae, you have succeeded. So both me and then a couple people in the chat have said, how can we follow up on this research that you have done?
So yeah, maybe next year at this time,
Yeah.
once we get all of that work done. So we're planting next month. We'll be harvesting the fall. There's going to be a lot of lab processing and DNA sequencing, but I'm hoping my students will be ready to present at a scientific conference next spring. And so maybe I'll bring them on and have them talk about it.
That would be fabulous. Well, plan on it.
Yeah.
And in the meantime, I hope your research goes well. And again, thank you for joining us. I know you may not be around for the second Q&A. You have some young, fresh minds to educate.
Yeah, I'll stick around as long as I can, but I do have to leave at maybe 10 till 1 to get to another event. But
All
yeah,
right.
thanks for the invite. It was great to kind of talk about this stuff.
Fantastic. All right. Well, Camilla, I'm going to pass it over to you. And again, thanks for joining us and looking forward to hearing your building on what Jonathan presented.
Thank you. Let me get the slides going here. Okay. Thank you so much for having me here today. I will be talking a little bit about the work that I have been doing with Jonathan here in Asheville area and with the American Chestnut Foundation. But before that, if my slides help me out here. So I will change a little bit the topic. Jonathan and I, we work a lot together. He's the plant guy, I am the bug person. So we try to combine our efforts and have a good answers for big questions. So But before that, before I talk about the projects that we have been doing together and the results, I would like to talk a little bit about my background and explain how I end up working with chestnut plants. So my background is in agriculture, more specific agriculture engineer, and I have all my training with biological control of pests. And when I was in New York working with Cornell, I was invited to talk with one of the growers there that was having a hard time with one of the pests. And I decided to explore more and trying to figure out how to help. And this grower was a organic grower who was having a huge problem with the chestnut weevil and his orchard, and was asking for some solutions that he could continue as an organic grower, but be able to produce enough. So the situation was very bad in a point that 1 year he lost more than 90% of his nuts with the weevil. So because of that, I didn't have much experience with the chestnut plants. And I sit down and start doing some research and trying to figure out how to support this grower. And I didn't find much information the way that will be helpful. Okay, you can use this or that, and this is going to get the solution. So I start doing different projects at the same time. And this is the way that I separate this. So I'm going to talk a little bit about those projects that make me have some better knowledge about the insect parts and how to try to do some biological control. And then when I move to here, how I change a little bit the focus. Okay, so first, when I start having the work with the chestnut, I start working not just with how to control, but understand the insect behavior. So to do this, I did at the same time 3 big projects. One, when I was exploring the phenology and how to check those populations on the plants. The second one, trying to figure out integrate pest management that the growers could use and be able to produce the nuts that they need. And the other one is check that what we was doing in our area will be helpful for other areas. So we start doing some collaboration with other groups who also study the chestnut. So we have these 2 publications that I'm going to talk really brief about some of the results. That was very good support for us, understand a little bit better. And we can see that this is already working for some of the growers. Still have space for improvement, but at least it's something that they can use already. Okay, those are free papers. If you want me, just send me an email. I can send to you those papers. So first one, we trying to figure out what's the best way to monitor them, what's the best way to track them, and what's the correct time to do the control, right? So we didn't know much about it, was a lot of talking, and the group who was talking in Michigan was in a different timing from the group in New York. So we had a hard time to figure out what's going on. So we decided to do an experiment and figure out the life cycle in both places and compare this and see what's going on. So after we do that, we also at the same time that we was trying to trap them and collect samples, we did on this picture here, you can see some red ones, more yellowish, the brown ones. Those are nematodes, larvae that we inoculate with specific entomopathogenic nematodes. Those are nematodes that they are specific, develop inside the insect. They are amazing biological control, but some of them are very specific. So the idea first is figure out some of the species that will be more intense and better control the larval stage. And then after we decide that, we trying to figure out the best one to apply on the field. So this, the first paper that gets out, it's about how we control them, and we test different combinations. First, we did the preliminary test in the lab, and we figured out that the Sternenema carpocapsae and Heterohabditis indica, those are the ones more efficient and be infecting the larva stage and stop the development process of the larva and kill them. But the big surprise for us is that, like all types of management, it's not just one solution, right? We did a combination with entomopathogenic fungi and also entomopathogenic nematodes, trying to get the life cycle in different times. And we was very happy that this works great, because we was able to see that the presence of the entomopathogenic fungi reduced the population of adults. And I'm going to explain a little bit how we put that. And the entomopathogenic nematodes was very efficient when the adults start getting outside the soil and start crawling to the trees. So the combination of the two was the best treatment. And we believe that this happens because they achieve the population in different stages. So after that, we saw that the population in Michigan has not completely, but was very different from the population in New York. And we was concerned that maybe we was not dealing with the same species, since that they have different timing, different behavior, different morphologies. So we decided to do, as soon as I moved to here, I had the chance to work with Cassius, one of my undergrad students, who did the genetic tests on both. And we figured out that was exactly the same species, but adapt really well for different areas. So this was something that we was expecting be different species already, and it is not. Was just amazing adaptation that they had. And this includes different color, different size, a little different leg shapes that they have. So small characteristics that for us entomologists, it's very important, and we was expecting to be different ones, but end up being the same thing. So especially with Casio's work, we was able to publish. He did some presentations, and our first presence with the American Chestnut event, where he was able to present some of his preliminary results. That was great. A great opportunity for him, and great opportunity for us, since I was just moving to Asheville, be able to know better and understand better the foundation and the program. That I was very excited with that. So then I move on for the part that I'm start working with the American Chestnut Foundation. I would like to highlight that for me, it's a pleasure to be part of this and be involved on this. And I'm always happy to be doing different work with the foundation. I think it's very important, the work that you all are doing. And I'm glad that I can use some of my knowledge to help out on this. So the project that I worked, and this is part of the same area that Jonathan was doing his work with, physiology. I tried to understand the invertebrate population, what's happened when we have this hybridization going on, how much this impact the environment, the invertebrates environment, the biodiversity that we have. To do that, we did experiments on checking the biodiversity of invertebrates above ground and below ground, also on the leaf litter part, the position. So this involves a lot of students. Some of the pictures that you can see here, those are my amazing students. Some of them already graduated and are working. Other ones is still here and working with me. So This one was one of the works where they collect leaf litter samples and also soil samples at the same area that Jonathan explained really well, the space there with the Dupont Forest group. And this one here, it's a funnel that we use to collect those invertebrates from leaf litter samples. And this entire structure here was this student who built that by herself. Of course, we help a little bit, but was all the design and everything for us to be able to run our samples. So on this idea here, it's exploring all the organisms on the leaf litter and also below ground and compare this diversity among the hybridization plants. Those are more experiments. We did some experiments where with the soil samples, we was able to do some traps when we are able to collect some entomopathogenic nematodes that was already on the area, that it's a good sign, right? Because if we have that, we maybe have some nematodes that it's able to control pests already on the area. So we was able to find some of them. This is the trap that we use. And those are some organisms that they was able to collect. Beautiful, cute ones. So some results for our surprise. Well, not much surprise. We always expect that and happy with that, especially in a forest environment. We was able to find entomopathogenic nematodes in all the hybrids plots, 75%, 94%, and 100% American. So the number of arthropods that we find was a little diverse and change a little bit over the time that we collect. This is something that we already expect because we have the life cycles for each group in different times. Arachnida, normally it's the highest ones that we find here, right? A lot of arachnidas on the group, especially on the end of the season. But 9.4, I'm gonna show more results about that, but 9.4% American chestnut was the ones that we found, for example, less Myriapoda abundance. This does not mean that we couldn't find that. With these results, we did not just abundance, but we check the index that compares the diversity that we have and also the numbers that we have in each one of the groups. And based on that, we was able to see that we didn't have much significant difference, just a 9.4% American chestnut, a little lower. We are thinking that this could be maybe based on the environment that we was collecting those. We had other things influence, like a lot of ants in the area that could influence these results, right? And this is gonna move me, please keep this in mind for the next experiment that we are planning to do in the future. So those results, we was able to go to the conference and they did the presentations, they did poster presentations, and they was able to have the training. This is something that I really like on this because it's not just the research, but also the training that we are doing for the next generation and how engaged they get on this topic and be involved in learning more. So this is another student who also participate on all the projects involved on that summer. So Evan now is working in Florida, just graduated too, and he did some evaluation above ground and also the herbivory part. So he found very cool results just checking the biodiversity that we was looking for. He did some testing on checking the Asian chestnut gall wasps, the parasitic wasps that lays the eggs and the amount of galls that increase there. He did some traps on the area and also measured the plants together with Dr. Horton now to compare this with the results that he found. So was a big project for him to do, and we have support the NEMA lab, our lab, helping out, but was him coordinating that. I'm not going to talk more about the site. I think Jonathan was really good in explaining all the area. So for his work with it, a similar index, right? When we check the Simpson and Shannon, those are just different ways to calculate, again, the type of species and also the number on each one of the groups, and did some statistical test for each treatment. So again, the abundance, the diversity, we found a huge amount of hymenopterans. Those are the ants groups, right? And this, we think it's the reason that we have highest. Here we found a lot of ants. We count a lot of ants. So those are examples of the pyramid trap that we use to collect some of the above ground. Those are more for crawling insects trap that we have been using to monitor those organisms. So this result shows that we don't have much difference, that it's a good thing, right? Comparing those hybrids, we didn't find big difference in index biodiversity. The amount of organisms was very similar in all the plots with different hybrids there. That was very good for us. About the herbivory, we did, and we adapt. This is a box that we adapt to be able to collect good pictures and do the measurement of the damage on the leaves. So we use BioLeaf applications. Those are specific apps that we can use, and they do the measurement of the plants and also the damage part. So based on the difference of that, we was able to calculate the foliation part of the experiments. And oops, just a second here. My Zoom was on the way here. Okay, so as you can see here, we didn't have much difference on the defoliation part, right? And the presence of and absence of the number of damage. It's a little bit different, but not significant. Okay, so herbivory was similar, and the hybridization, similar levels of herbivory. That means for us that we didn't have huge impact with hybridization on the amount of damage of the plants. With the Asian chestnut galls, this was a little bit different, right? We had a hard time to figure out the best way to do this approach, and this methodology was a lot of back and forth, going to the field, collect samples, not enough, and go back. So we come with good way to do this procedure and counting those and get the infestation in like a rank levels. I have all these details on the paper that I'm going to show that explains how percentage was the galls on the plants. We see on this one a little difference between the 75% and the 100%, also the 94% with the 75%. So a little difference on the galls, right? The number of galls in each one of those treatments. But again, this was one summer season and one specific area. So I would love to explore and do this in other areas when we have more trees in the area. So both assess show significance difference. One thing that we think influenced a lot was the age and the size of the plants, right? Because some of the 100% American chestnut was very tiny little ones, and the other ones were huge. So this, for sure, it's influenced the galls formation. And will be nice to do this exactly the same experiment in areas that we have same age groups of plants. So this is some conclusion and that we was able to see. So we get the insect community and the herbivory was not affected by the hybridization, but we had some difference on the chestnut galls wasps in different one of the hybrids, but thinking about that could be influenced by the size of the plants and age. So this was another publication. As you can see, a lot of students involved on this and training using this project as a very good opportunity for research and educational as well. So another project that Evan was helping out, but who is in charge of this project was Harley. It's a project where we have been working and dealing with having the larva, and one of the projects that I did in New York was measurement how many larvas we have inside each nuts, right? This could be influenced by the different hybrids that we have. So we start doing more specific experiments and figured out if the amount of larvae in each one of the nuts is going to be changing over time, and this is going to change between hybrids as well. To do that, we did some experiments when we evaluate the nuts almost daily for 2 months and trying to see what's going on, how many larvae was, weight, everything. And this is coming soon. The paper, we're still working on it, but we have good results showing the difference between the hybrids in weight, because the nuts was in a different size. And this is in partnership with the Mountain View group there. So it's more specific with the hybrids that we have better control of the situation and the size of the plants. Together with this, we decide to explore more. I have a chemical ecology background, and we decide to explore more how what guide those larvae to get outside and get to the soil. Because if we can understand what's going on, it will be easy for us to figure out some management approach for this. So this is a project that we start, and unfortunately, Harley just graduate right after. So I'm still holding, and I'm gonna probably start doing this again this summer, is where we create like olfactometers chambers, and we test the behavior of those larvae and different approach, different ideas where they want to go, different clues where they go, how they move, what they use. Could be roots, could be a specific type of soil, how they move through the soil to be able to pupate and get to adult age. So this is a cool experiment that is going on, and I'm waiting for having more larvae. I know a lot of you don't like me to say that, but I can't wait to see the larvae coming to the lab so I can play with them. So this was also a small publication. We tried different ones and already different things, and until now it's not working. So we are gonna keep trying different things to attract them or repel them. I hope soon we can have some solutions. The paper that the information about the experiments that we did with the American Chestnut Foundation, we was able already to publish my part. I know the part with the physiology is coming soon. It's under review, but the paper regards with the invertebrate population biodiversity, it's available as well, and have all the information about the impact of the hybridization. Now I would like to share some of things that I'm planning to keep doing and working, and I hope together with the American Chestnut Foundation. Before you guys say I didn't apply, it was my fault that I missed the deadline for the application for the future projects, but I will be working, and next one I'm gonna be submitting some. But the idea is keep checking the insects community above and below ground. Similar experiments that I was doing at the DuPont Forest, but now at the Mountain View farm, so I can have better control of the situation there and use this for pollinators and other insects. So the idea is been working with the hybridizations, right? the different groups of hybridizations and do all these methodologies. So I'm going to show you some pictures of those traps that we have been doing in other areas not related with chestnut, that it's my goal to start using with chestnut right now. So the pyramid trap that we use before, but be more efficient on the areas that we're going to use. The pan trap, this is really good for checking pollinators in the area. So I'm planning to install some of them. We did that last summer as a pre-test, but I'm planning this summer be able to add some of them and be checking more about the pollinators around the plants. The leaf litter and the soil samples, we are good to go to have more samples this summer and get a better understanding what's going on between the hybrids. The herbivory and also all the other ones that involves the weevil situation, especially the behavior. It's something that I would like to explore better and see the galls if it's the same situation as well. So I know it was a lot of information. My idea was just doing an overview and make you guys have a lot of questions for me. So I may be able to help out and answer those questions. I just let you know if I don't have time to answer all those questions today, I will be happy to answer through email or have meetings and talk about this in another time. Before I finish, I would like to acknowledge all the group who works on this project, everyone who is involved on those results that I show until now, especially the group who is working with me in the lab. I have a fantastic lab with amazing students that I'm very proud of them. They do research as a master's and PhDs, and it's the only way that we're able to generate that. So thank you for your time, and I will be happy now to answer questions.
Great job. That was fantastic, and is some information I was really hoping you would get to today. So before Jonathan has to leave, because I think we only have 5 minutes for him, I did have a couple more questions, I think, on the mycorrhizae thing. Yeah, so oh, I just lost it again. Dang it. There we go.
Yes. Sorry, my computer glitched there. Can you repeat that?
Yeah, I haven't asked the question yet, so we're
Okay.
good.
Everything just went black for a minute. I wasn't sure what I missed.
Yeah, Matthew Huang asked, have you seen American chestnuts respond differently to canopy clearing events compared to Chinese chestnuts?
I have not. I've not done a lot of work with pure Chinese chestnuts. The only thing I've done is with those 75% American. And yeah, I don't really, I mean, I know I see a lot of Chinese chestnuts around because they've been planted. You'll see them flowering soon, but I've not really done a lot of work with them.
And I haven't seen that either. I think it's a really good question. I will say there's 2 plots where you can see some interesting Chinese chestnut regeneration, both in Virginia. One's outside of Blacksburg. And if Matthew, if you follow up with me, I can give you the location, but I don't know it off the top of my head. The other isn't pure Chinese, but it's Lausanne State Forest. There's a really interesting planting of 10,000 mostly Chinese hybrids, so like maybe 67% Chinese chestnut, American chestnut, BC1-ish. Trees that were planted from the Connecticut Ag Experiment Station. They were planted in the late 60s, early 70s, and there's no regeneration underneath. And so the question is, if we go in and we do that clearing, what kind of response are we going to see? Are we going to see regeneration happen underneath? How does that affect by that release of the area? So I think that we might have opportunities to look at that question. One more question for you, Jonathan, then I know you got to go. Marcia Case asks, I purchased some mycorrhizae fertilizer. Is this the best way to encourage it, or compost, or what?
So a lot of those mycorrhizal fertilizers are blends of different spores from a bunch of different species. And I think mostly they're for gardens. And so they're mostly the endomycorrhizae, which is a different type of fungus. In terms of helping with compost, I'm not sure. I think that would be more some sort of saprobic fungi that are more decomposers. So, but I've seen those like mycomix things that gardeners can buy. And a lot of that, like I said, is more of the endomycorrhizae stuff, which is more common for mycorrhizal associates with herbaceous plants.
We often recommend that people get like some duff around an American chestnut, or even
Yeah.
how do you feel about that?
Yeah, I mean that's a good strategy because that would be supporting a duff or soil because that would be supporting the fungi that are around that are being supported by that tree.
Okay. Yeah. I think there's an opportunity for a Simpsons reference here to have like Duff branded Duff mycorrhizae for those who are old like me.
Well, the Simpsons are still around.
Yes. All right. Well, thanks, Jonathan. I know you got to go. I'm going to switch over to the questions for
All
Camilla.
right,
You have quite a few. Camilla Matthew Huang asks, how did you trap the Cisai samples? I'd like to know more about the methods you used.
Okay, so one of the papers that I did the citation there have some, let's see which one, I have those open here. I think was the one with the manage and biological control. I have including pictures of the traps that I use to get the adults. And for the larva stage, what I did, I put buckets in the soil with holes, and I create like a small microcosmos to check them and get the life cycle. It was an experiment, 2 years experiment for me to do that. But for monitoring, we test 3 different methods that it's used for different weevils, and the best one for our case was the pyramid one, the one that I show in some pictures that you can, it's a commercial one, you can buy them, and they simulate the trunk tree and the insects just crawling and get stuck on the container on the top. So this is the way that we monitoring the adults. The larva, it's the project that we are doing and we are planning to do this summer. We get the nuts before the larvae start coming out. And when the larvae start coming out, we have individual containers for each one of the nuts. And we collect daily every single larva that comes outside the nuts. So it's how we are doing the monitoring for them.
Great. And a plug for our May Chestnut Chat. We have a couple of researchers from the University of Memphis who are going to talk about their work with weevil as well. So there might be some hot weevil tips in May for folks who are interested in that. Mark Carlson asks, what fluid was used at the bottom of those funnels to preserve the specimens?
Ethanol. So the way that we preserve them, it's ethanol 7%. Most of the time.
70. Yeah. Okay. Troy Hallman asks, are Myriapoda generally beneficial or detrimental? Asking because 94% hybrid seems to have a significantly lower population compared to 50 or 100%.
Yeah, so they are unique group that they are very important for nutrition cycle, right? And we need them to be there. But it's tricky to collect them, especially with the traps that we use. So I'm not super concerned about the Myriapoda group at this point.
All right, that's good. I'm glad that Mike Alcott is on here. Hi, Mike, and you have a great question, if I can find it, regarding leaf herbivory. Spoiler alert, Mike also did a leaf herbivory study, and it's in the Chestnut magazine. Did you have any data on the species of insects that were eating the leaves? Could it be that non-native insects like spongy moth prefer the hybrids, while native insects prefer the American trees, but overall, the total herbivory quantity is similar?
So good question. And I would like to talk more about that because we didn't do a specific correlating with what type of insect was doing the herbivory because we was not able to collect at the same time the insect was eating. We just collect the leaves separate from the insects, right? So we couldn't tell exactly who did the herbivory.
Yeah, and I think that's definitely something to follow up on, and I hope.
Yeah, I think it will be great to talk more about it, and maybe we can come back with some specific methodology to do this.
Yeah, awesome. Rachel Cannon asks, can growers obtain the same strains of fungi and nematodes which were effective in your study?
Yes, not all, but the ones that I use for the field experiment, they are all commercial nematodes and commercial fungi, and all the names and specifications is all on the paper too. So, Beauveria bassiana, you can pick the brand that you want, right? And the entomopathogenic nematodes is the tricky one. I have been working with a lot of partners because they sometimes is really good, but the transportation or something happened and they arrived all dead. So you're gonna apply dead nematodes that is not gonna help out. So has a space there that we can talk. This is a completely different topic, but how to use those nematodes as a biological control is another thing that it's very important to talk about it.
Yeah, nice. You're also gearing up for a repeat event here, right, Camilla? I mean, coming back, maybe we make it a regular event.
We can do that for sure.
Yeah,
I love to talk about those things. I love what I do. So
that's
yeah.
great. I love to hear it. Debbie Milks. Hey, Debbie, thanks for coming and joining us. She has a question. She has found some codling moth larvae damage in her chestnuts. The moths were identified by Michigan State. Have you found codling moths in your collection of bugs?
I need to go and review all the bugs that we was able to identify. I can't tell you right now what's going on there, but we found some moths. I'm not sure if it's the specific one that you're talking about. Yeah, but I can follow up with you because we have all the ones that we did the identification already.
Okay. John Hempel says pan trap is, I guess, like fly paper in a pan? Question mark.
Pan trap. No, pan trap is the containers that I use it with different colors to attract, because a lot of the pollinators are guided by color. So we use those with different colors to see the diversity that we have in pollinators in the area. So it's not pan, no.
Okay.
It's a container with water.
All right. I'm glad we clarified that for him. Eric asks, if I have weevils, what is the best way to get rid of them?
They're so cute. Why you need to get away with them? No, send to me so I can do experiments with them. Well, I have different techniques that a lot of people are using to avoid the weevils get outside the nuts, right? And I always keep playing that those are extra proteins, right? When you cook and put inside the nuts, it's an extra proteins, but we are trying to avoid them to get inside the nuts. So my big goal here as an entomologist is to figure out a way that we can have a good production without have the larva inside the nuts.
And there are a couple of jokes here, and Dominic, you know, did the most common one, which is if I had to choose a weevil, I would opt for the lesser of the two weevils. That's always a good one. And I don't know if Tim X on here, but he even has a t-shirt. So Dominic, if you're looking for a sweet t-shirt, I can hook you up with Tim X and maybe he can get you one of those.
I would like one.
Would you? All right.
Yeah.
I'll see if I can get you one. I think from our standpoint of what we recommend for growers with weevils, we recommend really good sanitary protocols. That tends to be the best way. Commercial growers will often spray, and they spray like really heavy duty pesticides to remove the weevils. Unfortunately, there's a lot of secondary damage done to beneficial insects as well when you do that. But I mean, they're trying to make money, so you know that tends to be how they do it. If you're a backyard grower and you can handle it, get up all the nuts, get up all the burrs, do it as quickly as you possibly can. Burn it, because what happens is those weevils, they exit the nut, they burrow into the ground, they hang out for a year, then they come up. So the more you can reduce those larvae going into the ground, or pick them up and send them to Camilla. It sounds like she's willing to take those. You know what you could do? People could like gather all those nuts, put it into, I recommend steel because they can chew through plastic. Put it in like some sort of metal container, let them crawl out, and then they can send them in your refrigerator. And then
So yeah, I was kidding about that, but the reason for that, all the chemicals that we can see, it's not working effectively on the control, right? So at this point, we don't have a good control. Doesn't matter if it's organic or commercial one, we don't have. And the best way it's avoid the larva get outside. So when we leave the nuts on the ground, we need to clean everything, right? And make sure everything is clean and avoid that the cycle keeps going. We need to break the cycle. It's the idea. So I was able to show that we can use the entomopathogenic nematodes. We can use the entomopathogenic fungi. That was really good, right? And the growers that is using, they still using that. They maybe do some rotation with type of fungi, with type of entomopathogenic nematodes, but has been working really well for them to do that because attack the cycle in different stages. It's what we want, right? And understand what's going on in your specific area, I think is the best approach to get better management. But for sure, clean it up the area. It's a big deal.
Yeah. Dan has a couple good comments in there. Everyone, if you can check out some of Dan's in the Q&A. He says, eat your weevils, fry them up and use them as croutons, that they are very
Extra
tasty.
protein. Yes, just saying.
Yeah, and he says, again, boron. He recommends boron for weevils. Well, I think we can stop there. That was an excellent presentation. I really appreciate everybody coming and asking these great questions. Steven says chickens love the larvae, and that's what I hear. Guinea fowl, chickens, things like that they'll eat the larvae. So April, again, we're April 18th, we'll be doing the chestnut growing town hall. If you want to try and be a panelist on here, please let me know, and let me know what your growing tip is, and we'll open that up. May, we'll do follow up on weevils in the eastern US with Dwayne and Michael from the University of Memphis. In the meantime, I hope you guys have a wonderful spring. It looks like tomorrow is going to be like 80 here in central PA. So spring is certainly trying, and even summer might be trying to hop over spring here right now. But Camila, thank you again. That was really fantastic. And for, all of your ongoing support of the work and the research that's really important to what we do from a restoration standpoint. So thank you.
My pleasure.
Yep.
Thank
I
you
look
so
forward to
much
seeing
for
you
having
soon.
me. Bye-bye.
Take care. Bye.